Corona something. Can someone read it? You read it, Sarah. I nominate you.
Important update from.
That is so pleasant. Easter eggs in. What's that
Vinegar?
called? It's not Easter yet. It's still St.
Why did I not
Patrick's
see
Day.
this either?
I didn't see it either.
For what? Maybe it's only she got it. For what?
Alright, smells nice and tart in here.
Monday, Tuesday, Wednesday. Oh my god.
Yeah.
Wednesday, my dudes.
Alright.
There's that laugh.
I'm gonna do it.
Yes. That laugh. It's a go-go laugh. I don't think so. That's a real laugh.
We have some good laughers. Who's got a good laugh? Noah, senior Noah. What's her last name?
Know that?
I love her. Her laugh.
Uh-huh.
I love my pen and my skinwalker.
There's something about his that sounds like a little evil.
Oh, like he does that on purpose. That's not his natural.
I don't know if it's, I think it's pretty frequent, so it like frequent appearance. Alright. Someone knocking? Alright, so hang on, a few things of orders of business, right? I invited you guys all. Did you notice that I invited you to a Google Hangout?
Wait.
It's a joke. And it's not a social occasion.
That's like the ghetto Samsung FaceTime.
So anyway, what?
Like, come.
I'm not allowed. Okay, so we're like actually legally not allowed to like interact with you guys on social media. So I can't So I invited you from my school email to this Google Hangout in case we get closed down for some, because health, public health crisis. You guys need to make sure you check your Google Hangouts. Make sure you can access. Does anyone not have internet in here? Right. So you make sure you can access the Google Hangout, right? And apparently you can. So we can all be on this it's like a group chat, right? We can type on it. You can FaceTime on it. I tried it this morning. I don't know how it works with multiple members.
It's just like, okay, so like
Do you have all the people's faces on
yeah,
there?
but you can click who you want to be the biggest. So you have the biggest, and then you have just a bunch of little tango squares.
Okay, so we could do that. That would be kind of cool, and anyone can talk whenever they want. And so it's just like a group, conference call. I can also kick people out of it. Apparently, I saw you can remove someone from your group. So I didn't say I would, but I said I could. So I can hold that power. All right. So just that that's like the plan in case something happens. What would happen is we can do that group chat, right? And then I can share my screen, I think, with you guys.
Oh, yeah, you can.
So if I opened a PowerPoint or something, I could open, I could share the screen. You guys could make my screen the largest. I hope that you would make that screen the largest, not someone else.
I'm pretty sure that you can look at yourself too.
I can look at myself?
Yeah.
Oh, you can have yourself be the largest. Okay, so, all right, and if you're really, vain and want to look at yourself as the largest, that's okay.
You know what you look like.
So you know how you look like. So maybe I would do that. Oh, I would do that. Okay. All right, so I would share my screen with you guys, right? I would share the screen, and then I would just talk, and we'd go through the PowerPoint together. You guys could ask questions as normal, and then I would post the worksheet. And so it would be kind of like that. Obviously, we couldn't do labs or anything. I might assign you virtual labs to do. There are websites where you can actually perform the lab virtually. That when you're in desperate times, we have to, settle for certain less than perfect conditions, all right? But you guys all know how to do that, so I wouldn't lose contact with any of you guys should there be some sort of
Oh, she's gonna get about an hour.
interruption in class time, right? Okay.
go to the bathroom, you have like raise your hand in the Google Hangout.
You just get up and go and just record.
Yeah. If you raise your I mean, I honestly don't know how it works. I guess you guys would just have to sort of be cautious and like if you want to speak, you can. I mean, I would feel like see you guys, right?
Tap on the camera so that it flashes.
You can what? Tap on? Oh, tap on and off.
Or wait, just like get out your phone and record it, and so like you can like go back and listen later to see what you missed and just quit. You know.
Oh, that's smart. You can screen it.
Oh, if you have to go to the bathroom.
No. So you can.
I'm glad you guys are preparing for all these contingencies. So.
What am I going to be?
Or you can just like go on mute and turn off your camera, and then you can just go and like.
I mean, whatever business you guys need to do, you know, it's the same thing as here, right? When you get up out of the class and go to the bathroom, you
You whisper to you.
You can eat food. I mean, yeah, it's not going to be like messy in the lab or anything. But like, I prefer that, like, yeah, if you're gonna eat food, you know, you don't need to like make a show of it. You can just be, you know, like eating politely. I don't know, whatever. And you guys saw the schedule, so there would be an alternate schedule. It'd be like only 1 hour each day, right? And there would be like 3 in a row and then a break and then the fourth, I think. So, what? I think the break is like 20 minutes. I don't really know, but you know, anyway. So we're all aware of this possibility, right? And we'd all be ready to, you know, to still interact online, right?
If you're sick, do you still have to go to school? Like, what if I'm like in a sick day?
I don't know how that works at all, actually. So, huh? If you have coronavirus and you're like safe in your home, unless you're like, you know, sick and like not feeling well and unable to like, you know, actually look at it, or if it makes you dizzy to look at the, you know, I think you can still participate even if you have everyone at your house. Because honestly, the symptoms in younger people are pretty mild, right? So, I don't know. All right. So here's that's not what Miss Jolie said. No one is dying. I mean,
It's not so simple. It's
Apparently, like 80% of Americans are pretty considerate.
Yeah.
yeah, look at this.
when I was when I was
That's almost worse than CNN.
when I was young, do you guys you're all vaccinated against chickenpox, right? Yeah. So yeah, I think you guys have to be varicella, right? So, but when I was younger, maybe this shows like our generational gap, but we were supposed to, we weren't vaccinated against it, and we actually, like if someone in the neighborhood like was like, you know, like rumored to have contracted chickenpox, they would send all the other kids over and have like a chickenpox party so you could get it when you're young, and so you can't get it again, and because it's way better like when you're young to get it, right?
There's an episode of Peppa Pig where that happens.
That's what my mom did.
Yeah, mine too. Peppa Pig, where like all the friends, like Peppa gets
Chickenpox, and they all send them.
What's on her?
All right.
She has chickenpox.
Yeah.
I didn't know we were supposed to type it up.
I don't want chickenpox.
All right. Some people didn't share it with me last night. The people who did share it with me early enough, I like commented on it, and I kind of like made some points for you guys to fix, and you did, and thank you. That was good. Huh? Like before 11ish. I don't know. I was like on my computer and I just saw them pop up, so I was just like, you know, I wasn't like picking on you guys, but I was just like, oh, okay, I'll like point out a few things here and there. I didn't get to do that to everyone because there wasn't just enough time. But we're gonna go over it today. So if you notice that there's like a big discrepancy between like what we're describing here and what you researched, you should be like a little concerned.
Sorry, this is on the topic. Are we getting our test back?
Yes, and I'll give you your test back. I think we have enough time. I think we're not going to start the models today. We're going to do our DNA models tomorrow. But so today we're going to do the history. We're going to do the structure in general. I'll give you the 2 worksheets, right? And then you can complete that for the rest of the time and for homework. And then I'll give you your lipids quizzes back. Were you guys happy with those or not?
No.
Okay.
I was.
Some of you guys might have been, some of you might not have been. I wasn't like completely convinced that everyone completely understood it based on like so I do want you to go through and like mark the correct answers or try to find the correct answers for the ones that you got wrong. Right? I also noticed that like when I was looking through your carbohydrates quizzes, the ones where you corrected the answers, I noticed that a lot of you guys corrected the answers to another incorrect answer, which is also a little concerning. So I think I need to, like, I'm going to check. So I'm going to give them back to you guys. You should correct your answers, and then I'm going to check the corrected answers. Okay? All right. So let's get through these experiments. I know it was kind of, was it interesting or was it kind of boring?
One of them was interesting, and the other one, like the rat one, was interesting.
All right. Some of them are kind of hard to understand, but we're gonna try to like tackle them together, and I think you guys can do it. So our objectives today, right? The main thing is that we're gonna describe those experiments, the 3 experiments for like what led to the discovery or that DNA was the genetic material. That's our main goal today, right? And if you guys still don't feel like by the end of today that you understand those experiments, you need to come see me or you need to talk to your tutors or something, right? Okay, and then we're gonna get into the structure of DNA if we have time, but I don't know if we'll get to that today. Okay, so the 3 experiments. What was the first one?
Griffith.
That was by Griffith, right? Frederick Griffith. The next one, okay, Avery, MacLeod, and McCarty, and then the third one was Hershey Chase. All right, so these were like the really like. I know it's like boring to go into the history, but they're really important because nowadays, what do we know about DNA? You guys wrote some of it, like some of you guys wrote it on the back of your thing. What's the main purpose of DNA? What's the main like job that it has? It carries genetic information, right? But back then, did they know that? No. And so like we really take that information for granted now, but like they had to work to get that information that we have now, so we have to like give them some like respect or some tribute, right? So by studying them, okay? All right, so Griffith was working with these 2 strains of bacteria. What kind of bacteria were they? Okay, so there are 2 strains of bacteria that cause pneumonia, right? There are Streptococcus pneumoniae, or whatever, right? So then there were 2 different strains. There was one that was smooth, right? And what does smooth mean? Smooth meant that on the outside of its coat, it was smooth. Why? Arya?
Because it had a protection.
Exactly. So this strain was virulent, which means harmful or like disease-causing, pathogenic, right? And it was virulent because the outside of the cells were smooth because they were surrounded by this thing called a capsule, right? Which is, do you guys know what that's made of? Sugar. It's made of polysaccharides, right? So actually it protects them. So if like my immune system wants to go eat this bacteria that's like invaded my body, can it do it? No, because there's that protective coating on the outside of this bacteria, right? So this one, the smooth one, was bad because it had that coat and the immune system couldn't kill it, right? The other strain was the rough strain, and this strain, what was the effect on the mouse? It didn't do anything. It was innocuous, right? Do you guys know these vocab words? Yes. Innocuous means like sort of innocent or like not harmful, right? And so I depicted them as like this one is rough. It doesn't have that smooth coating, so the immune cells can like easily just get it and no problem, right? Okay, so there are different stages. So the first stage, Griffith took what? Live estrine. A lot of you guys just said estrine some of you guys just said S strain or R strain, but I wanted to specify whether it was alive or whether it was killed, right? So he first took the live S strain, and the immune cells, can they get it or not? Can they attack it or no? No, so what's going to happen to the mouse? The mouse is going to die, right? And when we extract a sample from the mouse, what will we find inside the mouse's body? We'll find living S strain, right? That makes sense because we're injecting the living S strain. The living S strain kills the mouse, and there's still living S strain there. Makes sense, right? No surprises? All right, what if we have R strain, live R strain? What's going to happen there, Arsh?
The mouse will live because it's innocuous.
Good. It's innocuous because the immune system can kill it, right? So we're going to find, the mouse is going to live, and then when we take a sample from the mouse, is there going to be any S strain in there? No, there's going to be living R strain, right? Or maybe the immune system will have killed the R. So but there's definitely not going to be any S, right? We agree on that?
Yes.
Okay. Here's where it gets interesting. All right, so now, and I'm I don't know how to depict them, so I just made them look like kind of weird like fingernails. Kind of gross. But that's the point is that those are denatured because we heated them. So those are killed straight. So and again, these are all what are these? These are they're bacteria, right? So these are bacteria, those are dead bacteria, but they're still bacteria cells that have been just all like melted up because I heated them, right? So what's going to happen if I put the heated S strain alone into the mouse? Nothing. The mouse lives, right? Nothing. And when I extract the sample from the mouse, is there any living S strain in it? No, because it's all heat killed and like warped, right? Those like weird fingernails. Okay. But then the weird part is when I put, so this one didn't kill it and this one didn't kill it. But when I mix those together, what happens? The mouse does die. And when I take a sample out of the mouse, what do we find in there? Living S strain. So this is like the question. What happened? Transformation. Good. Do you guys all understand what that word means in this context? So transform means to do what? To change, right? But in this case, what we're talking about is, so this is what happened. Remember we mixed the live R strain and the dead S strain, right? And so what's happening? There's something physical in those dead S cells, bless you. In those dead S cells, there's some physical factor that these cells are like picking up from their environment and somehow like acquiring some sort of like trait from that dead material right there, right? So, and then by doing that, they're becoming transformed, right? So transformation, if you were to define it, would be, can anyone sort of make a stab at it? Arsh?
In this context, it's like almost like taking the DNA from the dead S strain.
Do we know it's DNA?
Well, I mean the dead quality that like makes it lethal, and then absorbing that for yourself to use.
Okay, good. So you're taking something from this, you're picking up something from your environment, incorporating it into you, and then becoming changed by that. So it's like picking up stuff from your environment and becoming changed. Yes?
But would that, would it be a modified S strain if it's still an S
No.
strain?
It's the R. Yes, it becomes the S strain because it has a smooth coat then. So basically what happens is, and again, sorry for the sad animations, but this is like my new depiction of it, right? So this is like the R strain that has somehow like picked up something that gave it like a shiny new red smooth coat, right? So that's the new one. But then what's the thing that really like sort of is the kicker here? Like, an individual, I could just like put on a coat myself, and that doesn't necessarily mean that it's a genetic thing. Then what has to happen in order to prove that it's carrying some sort of genetic information? George?
Like it has to actually take over its qualities, so it was able to kill them.
Okay, so it took over the qualities. But suppose I put like this like red coat on myself and then I kill the mouse. That's possible, right? But to show that it's like actually genetic, what is that?
I think it's like a transformation principle, like transfers the genes that kill the mouse.
Okay, but the genes can all. Okay, you guys are. This is all right. These genes are definitely being transferred to that one individual. But to prove that it's something that is like not just on the individual level, if I go like to the gym and like work out, like that doesn't mean that that's genetic. That's like more of like a change that I'm doing as an individual. Juliana?
It takes on like the actions that they do.
That's sort of like the same thing that she's saying.
Oh wait, I think I know.
Completely altered behavior.
Will your daughter also kill them?
Yes, that's what I want to see. Not only, my daughters could definitely kill me. Not only does that one bacteria that actually picked up the DNA, like the stuff lying around. So suppose these are like some like dead S strains, right? And then I'm like the R strain. I'm like hanging out, and then I'm like, oh, okay, like I'm gonna like have this now. Now I have all these new powers because I have this thing that the S strain sort of left around, right? So I myself have those powers. So that's what you guys were talking about. You're talking about like how they acquired those powers, those new traits. But it's not only that, but then when I have a child, my child will also have those traits. That proves that it's actually genetic material, right? So the fact that not only the fact that they picked it up from their environment, but that they also passed that along to their next generations, right? So that proved that it's like this genetic material, right? Okay, so what was the conclusion from Griffith? So before Griffith was like doing his experiments, people thought, how did they think that like genetic information got passed along?
Proteins.
They didn't know.
I mean, there was no physical thing that they were identifying. They thought it was kind of like magic, right? Like, you know, it was like they called them like, so even Mendel, you guys know who Mendel was?
Oh yeah, the deep guy.
Yeah, he was like that monk who like
German.
bred all the pea plants. Even though he like was making all these patterns and like figuring out all the rules for genetics, he actually didn't realize that it was something physical that you pick up and that like you can put into another organism. He thought that it was just kind of like this spiritual, like heritable factor. But now Griffith is proving that it's actually something physical in this dead S environment that's making them become a certain way. So the conclusion was, and this looks really long and scary, but it's not. So innocuous bacteria, which is which one? The R strain, right? Picked up a physical, so the live innocuous bacteria picked up the physical transforming factor, which we know now is what?
The DNA.
We know it's DNA, but he didn't know it was DNA. He just knew that there was this physical thing from these dead S cells, picked it up from these from the heat-killed virulent bacteria cells, and then they passed it on to their offspring, right? Which then proved that it was this genetic material. So this is the genetic material, right? So it's a physical thing, not like some spiritual thing. That's really important for us to know. Like, we take it for granted. But then they thought it was something like kind of like in the air. So that's why it's really important to know that it's like this thing in these dead cells that I'm actually picking up, right? Right. So you guys are really, so if I asked you all those questions about which is S, which is R, right? Which one is virulent, which one is innocuous, when is heat killed, which one's not, like which one's mixed together. So suppose I mixed like heat killed R with heat killed S. Would that kill the mouse or no? No, right? Why? Because they're, the R's not alive, so it can't pick up the genetic material, right? Okay, so suppose I put in heat killed R and living S. What's gonna happen? It's gonna kill it. Living S ever is always gonna kill it, right? And what if there's like heat killed? So the main tricky one is that if you have live R and heat killed S, you're still going to heat killed. Still going to kill. He killed S and live R. Jasmine? Transformation means picking up genetic material and then acquiring new characteristics from that genetic material that you pick up, right? Okay. Can I go on to the next one?
Yeah.
All right.
What is it? Picking up genetic material?
Picking up physical genetic material and then acquiring the traits that genetic material carries. And becoming transformed by that, right? So, all right. Okay, so what did Avery, MacLeod, and McCarty do? Was it related to Griffith at all, or no?
Yeah.
Okay, Arsh?
So what they did was they basically tried to figure out which material was actually being transferred with this transfer.
Okay, so good. They extended Griffith's experiment. And in these, so we have all these heat-killed dead S cells right here, right? What do they have in them? And you guys, we haven't really studied that much about cells, but what do cells have in them? Membranes, which are made of what?
Amino acids.
Phospholipids, right? So it could be, they have lipids. What else do cells have in them? Proteins, mitochondria, nuclei. So they have DNA, RNA. They have carbohydrates, right? So what is it in these things that I'm actually picking up that's changing me into a new bacteria, right? That's giving me this smooth coat, right? What's the thing that's carrying the actual information? So they want to know what it is in this soup of dead cells that's actually helping transfer that material. So they thought it could be, totally wrong page, right? All right. So they thought it could be the proteins, right? They thought it could be lipids. They thought it could be carbohydrates in that, soup of dead S cells. It could be RNA or it could be DNA, right? So in order to test which one it is, what do you have to kind of do? We do?
Isolate each individual part. So like you isolate just the proteins, just the lipids, just the carbohydrates.
Exactly. You have to isolate them, or you have to pretty much knock one out. So when one's not there, is it still being transmitted, right? So that's what they did, right? So they took, can I go to the next slide? All right. So basically there are these enzymes that end in A's, and they refer to the molecule that they, this is actually DNase, right? And then there's RNase too, right? I'll add that. And it's like, I think it's like,
I think the A is capital. That's
the
not
A is capital?
No, the A isn't capital.
No, it's not.
It's
No,
A
it's
is
not.
I saw it as not.
It's a lower.
I saw it capital.
We'll leave it like this, like both, alright? It doesn't really matter that much, but in any case, it's definitely not this, but you get the idea, right? The idea is that this thing gets removed. So if I put a lipase in, a lipase is an enzyme that gets rid of lipids, right? If I put it in a protease, that's an enzyme that breaks up the proteins, right? DNase is going to get rid of DNA, and then RNase is going to get rid of RNA, right? They don't, when you have an ase ending, it doesn't always mean that it gets rid of that thing, but in this case it does. Okay? So what did they do? So first they took what? And they remember they're wondering what's in here, right? That's making the change, right? So I have to take what first, George?
The heat-killed S strain.
Good. So I have to take the heat-killed S strain first, right? My weird fingernails, right? And if I want to see, suppose I think it's proteins that's transferring the information. What should I try to do? Put in protease and get rid of the proteins, right? And see if these cells, when added to what? When added to this is the heat killed, right? When added to the live R strain, right? Are they still going to kill the mouse? Is that information that they were carrying about having a smooth coat being transferred to the live R strain? Did it get transferred in the case when proteins were gone?
No. Wait, no, it did.
Wait,
It did.
oh,
It did.
yeah, it didn't when it was
It did, my bad.
Okay, so when there are no proteins in here, can I still pick up the genetic material and incorporate it myself? Yeah. Yeah. So they can still, so this still killed the mouse, and when they extracted it, they still found live S cells. So what occurred still? Transformation still occurred, right? Okay. What about when they removed lipids with lipase? And then what do they have to do? Add the live R strain, and then they still find live S cells, right? And then you guys get the picture. So when they remove polysaccharides, same thing. That they're still getting live R cells. They're still killing the mouse, right? What about, what's the other one? RNA? Okay, so when they removed RNA and they added the live R strain, then they're still getting what? Live S cells. But then this one, when they add the DNase and they remove the DNA, they only were able to get the R strain, or they had no S strain, let's say, right? No live S cells. So then what did they conclude then? Exactly. DNA is the transforming factor, or DNA is the genetic material transmitter, right? So basically they knocked out all these things and then added to the R strain, the live R strain, and then they found which ones still transmitted the information. Easy? Mm-hmm. Okay. This one is kind of like the trickiest one, I think. Did you guys find it that way or no?
I actually didn't find it that way.
Okay, so this is the last one, right? Okay, so let's just try to stay with me, and if you have questions, please ask as we're going, right? So the T2 bacteriophage. What is a bacteriophage?
Something that kills.
George?
Something that kills bacteria.
Okay, something that kills bacteria, but what's that something? Lidu?
It's a virus.
It's a virus, right? It's a virus that actually targets bacteria, right? Do you guys know what's the relative size between a virus and a bacterial cell?
Virus is much smaller.
Virus is way smaller, right? Okay, so the T2 bacteriophage is it's like a virus that infects bacteria. And basically, before I do this, I want you guys to know. I want you to look at this, right? This is the bacteriophage. This is what it looks like, right?
Yeah.
And I want you to look. It has a head that contains inside. It has DNA or RNA, some sort of genetic material in it, right? But it has DNA or RNA. And on the outside, what is on the outside of it? Sorry, I should have actually gone here. So on the outside of it, and all this black stuff, this would be like a protein coat. And do you guys know what that protein coat is called? Capsid. The capsid, right? And then the DNA is on the inside, and that's like the core inside the head, right? Make sure you don't confuse these two words. What's the the difference? Charlie?
The capsule is on the outside of the live estrogen
Which is what? Is that a virus or is it a bacteria?
bacteria. And
This is
then
on the bacteria, right? Good.
capsid is like DNA inside of the
DNA?
yeah, the DNA inside. It's not the DNA. Oh, it's the protein outside shell of the bacteriophage.
Bacteriophage of the virus, right? Okay, and this is what's a capsule made of? Is that made of protein?
Polysaccharide.
Polysaccharide. So make sure you really distinguish between these two so you don't get them mixed up, right? The capsule is on the bacteria, meaning the S bacteria, the pneumonia bacteria, right? And it's a polysaccharide, and then the capsid is on these viruses, and it's the outside protein, not polysaccharide, okay? Okay. So what happens is when a bacteriophage infects a bacteria, so here's the bacteria cell, right? And then here's the bacteriophage, right? Which one's bigger again? The bacteria cell is way bigger, right? So basically what happens is the bacteriophage comes in, it binds, right? And then it inserts, it looks like it's like pooping into it, but it basically inserts something that's going to reprogram, some sort of genetic information that's going to reprogram this whole bacterial cell and to make it start producing all the bacteriophage's babies. And like it assembles them, and then basically it explodes, and then they go reinfect other cells, right? Do you guys know this sort of life cycle of a bacteriophage?
Oh, yeah.
It's really kind of creepy. Does the bacteriophage itself enter the cell? No. So scientists knew that there are these bacteriophages, and they knew that they were putting something inside the cell, some sort of genetic material that was reprogramming the cell. So, and the bacteriophage is made of one of two things, right? Of two things, right? So it could have been either what that was going in there.
Protein.
Ro?
Never mind.
Arsh?
Alright.
All right. Let's raise our hands, but yeah, okay.
I was gonna say phosphorus and sulfur.
You're okay, that's you're right, but Jasmine? Okay, so they're trying to figure out what is when this thing is coming, like here's this bacteriophage thing, right? And here's the DNA. Is it putting this DNA in there when it's like, you know, when it's doing this like weird squatting action, right? Or is it putting the protein coat here? Is that stuff going in the cell, right? They didn't know. So they had to figure out which one it was, right? That was the main question that they were answering, right? Okay. What is what they and you said phosphorus and sulfur?
For like 2 seconds.
All right, so the question was whether it's going, protein is going in or whether the DNA is going in, right? Okay, so what difference between protein and DNA did they take advantage of? All right, Ro.
DNA had phosphorus, and then protein had sulfur.
Good. So DNA contains phosphorus and little, if any, sulfur, right? And proteins contain sulfur, but maybe not phosphorus, right? So do you guys remember what an isotope is?
Yes.
What's an isotope? George?
It's like different type of the same element or atom, and it has like different amount of a different mass number.
Mass number and
It's like a different amount of neutrons in it.
neutrons, right? And so what would the difference between two isotopes be maybe? So if I have carbon like 12 and carbon 14, or if I have sulfur 35 and sulfur 36, what might the difference between them be? Do they react differently with different, like do they bond differently with different things? Mike?
Yeah, because they might have a more positive or negative charge.
Only the number of neutrons changes between isotopes. So the number of, what's involved in bonding? Arsh?
Just the electrons.
Just the electrons. Still has the same number of electrons. So is it still going to do the same bonding as other isotopes? Yes, except this time we can track them, right? Because sometimes one isotope will be radioactive and one would not be radioactive, right? And that's exactly what they took advantage of here, right? So protein contains phosphorus, sulfur, not phosphorus, right? And then DNA contains phosphorus but not sulfur, right? So I'm going to take 2 batches of these phages, right? 2 separate batches. One batch, I'm going to label the proteins with what?
Proteins.
Proteins.
Sulfur.
I'm labeling the proteins with sulfur. Sulfur 35, right? I'm going to write sulfur. I'm going to label them with sulfur 35. So where are they going to be radioactive then? On the protein cone, the outside of their stuff, right? Am I labeling this one with phosphorus? With radioactive phosphorus? No, right? But the other batch, I'm going to label with phosphorus. You guys remember the number? 32.
Yeah.
All right, so I'm going to label this other batch with phosphorus 32. What's going to take up the radioactive phosphorus? Are the proteins going to take up radioactive phosphorus? No, just the DNA, because the DNA is the only thing that has the phosphorus in it. So where is this one going to be radioactive? Inside of the phage, right? Okay, are you with me or not really? Yeah? What? Okay. And so what do these phages do when they get mixed with the bacteria again? They inject something in them, right? So we're going to expose both phage groups separately to separate bacteria, right? So this one's going in, and then they would inject whatever the genetic material is, and these are going to go in, and they're going to inject their genetic material, right? Are you okay with that? Okay. So what are the So what they did was once they exposed the phages to the bacterial cells, then what did they do? They put them through a row?
Blender and a centrifuge.
They put it through a blender and centrifuge. So what happens to the phages and the cells? So basically a centrifuge uses gravity, right, to kind of like separate things based on their density, right? So which is more dense, the cells or the phages? Go ahead.
The pellet, the cells.
Okay, so the pellet is the thing that's like the hardened thing that forms at the bottom, and then the supernatant is like this like liquid kind of thing that is on the top part, right? So if I'm centrifuging my phages and my cells, the bacteria cells are going to land down here, and then the phages are going to land up here, right? So I want you guys to really analyze this. So in the sample that had sulfur, what was it, 35?
35.
In the sample that had sulfur 35, I had these phages, and they were labeled with the outside coating, right? The protein coating was labeled with the radioactive isotope, right? And the cells. So what happened? What was the radioactivity between the pellet and the supernatant in the case of the ones that were labeled with sulfur? Where was this one radioactive, and where was it not radioactive? So here's the pellet, let's say, and here's the supernatant up here. Right? Row?
I think the supernatant was radioactive.
The supernatant was radioactive, right? So this one had like the kind of radioactivity. Sorry, I should probably make this. And this one was not radioactive, right? So that shows that what was radioactive. What's up here again? This is the phages, right? And this is the cells, and presumably whatever genetic material the phages had inserted would have gone in there too, right? So this is not radioactive here. So that means that what cannot be the carrying genetic material? Proteins can't be it, right? What about in the case of the phosphorus? What was the number again? 32. Okay, in the case of phosphorus, 32. So here's the supernatant, and let's say here's the pellet, right? Which one? So the cells. Remember the bacteria cells are down here, and here's the phages are going to be up here. Right? Which one is radioactive in the case for the phosphorus? George?
The cells at the bottom.
The cells, this one was radioactive here, right? So what did they conclude?
That had to be the DNA.
The DNA had to be the thing that the bacteriophages were inserting, and therefore that's also the genetic material, right? Because that's the thing that's causing the cells to go crazy and change their behavior and everything, right? So, yeah, so this one, which one did they find the Which one did they find the radioactivity in the cells? The phosphorus, and what was labeled with the phosphorus? DNA so that they knew that DNA was the. Sorry, I have like some update here. Okay. So it's phosphorase, and then we knew that it was DNA, right? Easy? Okay. So, do you guys need to go back to that or no? This is okay? All right. So basically, all these experiments led to the conclusion that DNA is the carrier of genetic information. That's it. All right? Okay. Are you guys ready for structure? Do you want to stretch for a second? You guys all look kind of tired. Let's stretch for 1 minute. Right? 90 seconds? All right. But quietly, but yeah, let's stretch. And I'll pass out the worksheets actually, so you can start doing them on your own if you want. Who wants to help me?
I'll do it.
Thank you. There are 2 things.
Oh, there's 2?
There's 2, like there are 2 for each person, so.
I can do the second one. I can do the second one.
Okay, why don't you, thank you. Let's just do one at a time then. Here,
Right.
Omar can pass out these.
Okay,
So give me the second pages of all these. And you just take the first pages and make sure every single person gets one. All right, we're good?
All right.
Thank you. Yes. Okay, we're almost done, guys. And then tomorrow you get to make the models, your own models.
Wait, what happened? Omar, this one's yours.
Make sure you have one of each. One should start with number 31 and one should. Yeah. And so we have an early day, like an early closing on Friday. So yeah, we just we'll just do the quiz on Friday still. Is that okay? Yeah. Quiz on this, like this stuff that we're doing today, and then it's basically just this stuff that we're doing today. Yeah, it's gonna be a pretty easy quiz, I think.
Why? What are we doing tomorrow?
Tomorrow you're gonna build your model, so that should just reinforce.
It'll be like the experiments and then like the structure and that's like project.
Yes. And then next week we're gonna start doing a lab. I mean, if we're here, I hope we're here. Otherwise, you'll have to do it online. There's like a virtual version of it. You want to come to school or you don't?
It'd be pretty cool. You wake up at like 7:30.
Yeah, and just like crawl right up to the computer and be like, I don't have any more. Who didn't? Wait. Can everyone check and make sure you have only one of each packet because we're short one, I think.
Who doesn't have? I have an extra number 31 right here. Yeah.
Good.
You're welcome, Omar.
All right. Are we ready to continue? This part is the fun part because it's molecular. So no more history. You guys love it. You know you love it. All right. All right, so some of you guys on the back of your quizzes wrote that you like knew what this was, so it's good. No one's like totally in the dark. So it stands for deoxyribonucleic acid, right? You guys know what deoxyribose is, right? What is that? That's a. You don't know what this is? You guys?
It's a polysaccharide.
Okay, it's not a polysaccharide. It's a pentose sugar, right? Monosaccharide. Yeah, it's a carbohydrate. Deoxyribose is, right? But deoxyribonucleic acid is a whole different thing. But it contains deoxyribose, right? Okay. So basically a nucleic acid, that's a biomolecule composed of monomers. You guys know what that means, right? Right? The monomers, and we'll go through this in a second. But you guys know what that means, right? And it forms by, how do you think it forms? How do other monomers combine to form polymers? Dehydration synthesis. So this is like all kind of like the same like idea going on here, right? And what's the purpose of deoxyribonucleic nucleic acid?
Carry genetic.
To hold genetic information, to copy it, and to transmit it, right? So it's like all these different roles, right? To store, copy, and transmit genetic information. Good? It's okay. Thank you. Okay. There's still plenty of time. Yeah. Can I move on? And I'll post this. Okay. The monomer of a carbohydrate is a what? A what?
Monosaccharide.
Carbohydrate. Carbohydrate. Okay, is a monosaccharide. The monomer of a triglyceride is a fatty acid.
Triglyceride. Okay, I thought I said no.
The monomer of a nucleic acid? Nucleotide. There we go. All right? All right. I want you guys to remember how to do this. So what is this molecule right here? Deoxyribose, right? And then I want you to be able to number all the carbons, right? So, can you do it? All right, so where do I start? Right, and then I go here. How many are there? Okay, so 1, 2, 3, 4, 5. That's a carbon 2, right? You guys remember that that's a carbon 2. Okay. So there are my ones. I want you to find the 5 and the 3 carbon. Do you see them? So the 5 is up there, and then the 3 one is down here. And this is really, really important that you know these in DNA because you can only build on the 3' end. So it always goes, you're always building, I can only build on to this part. I can't build up from that part, okay? We're good? Okay. All right, so a nucleotide. So a nucleotide is what again? The monomer of nucleic acids, right? So there are 3 components. First one, what's that? The pentose sugar. In this case, is deoxyribose, but it could be ribose, right? If we're talking about what? RNA, right? So there's a pentose sugar. That's like the first thing that we put down, right? Can I go on? Yeah? All right. So at the number 1 carbon, where's the number 1 carbon? That one, right? At the 1 carbon, I want you always add a base, right? So I'm going to add it here. Right? That's the nitrogenous base. So that always comes off the 1 carbon. And then off of the 5 carbon, I want you to add a phosphate group. Right? So where's the 5 carbon? Over there. 154. That weird one, right? The one with the little, looks like a little arm or something, right? That one there, we're going to add a phosphate group. Okay, why is that all weird? All right, are we good so far? Okay, and now I want you guys to find the number 3 carbon. Which one is that? Okay, what group is attached there? A hydroxyl group, right? Technically, the hydroxyl group is not part of the nucleotide because what is it part of? It's part of the deoxyribose, which is already part of the nucleotide itself. So to say the 3 components of a nucleotide, what would you say? Phosphate, base, and not hydroxyl. You wouldn't say the hydroxyl because hydroxyl is part of the sugar. So we say the phosphate, the base, and the sugar, deoxyribose. But you don't include the hydroxyl as part of the nucleotide. But you should note that there is a free hydroxyl on this 3 carbon, okay? That's why we're doing that. But the main components are these 3, right? All right, so there's a hydroxyl group there. You guys notice that, all right? So here, we're going to review. What's at the 3 carbon of the deoxyribose? The 3rd carbon. Hydroxyl, good. What's at the 5 carbon? Phosphate group, right? And what's at the number 1 carbon? The base. Easy? All right. So here, now we're going to start creating what? Polymers, right? So here's our monomer right here. This is like your monomer. And this is called what again? A nucleotide, right? So there's, and this part again, bear with me here. So how are we going to connect the monomers to each other? Dehydration synthesis, right? You guys are familiar with this? So, and remember, nucleotides can only be added on to which end again? This one right here, right? So I want you to just sort of watch and sort of try to appreciate this. We're not doing the actual molecular thing here, but I'm going to show you. So I'm going to bring in a new nucleotide. Is it going to be built underneath or on top? Underneath, right? Do you see what I did there? So that hydroxyl group went away, and then I added a phosphate here, and this phosphate, and what else came out? Water, because it's dehydration synthesis, right? And here's another, what is this? Another nucleotide. So now we have a dinucleotide, right? And if I keep doing it, right? Did it again, right? And did it again, right? Which is the 5 prime. So now that I have this like kind of longer molecule, I need you guys to always be able to identify the 5 prime end and then the 3 prime end. And I'm using this word prime, which basically means like the 5 carbon end and then the 3 carbon end, but they call it 5 prime. This thing, you say that 5 prime, and this is 3 prime. I don't know why, but that thing means prime, right? So which is the 5 prime end? The phosphate group, because that's the 5 carbon up there, right? And then what's the 3 prime end then? The hydroxyl group, right? So the hydroxyl group is way down there, right? And in carbohydrates, what were the bonds that we made between the monomers? What were those called? Glycosidic linkages, all right? In fatty acids, ARIA, what were they called? Ester linkages, right? What do you think they're called here? Do you know?
Nucleic linkages, I don't know.
So this whole bond, and I want you to like, it would be like those right here. Those are called phospho. You get it? Phospho. And diester, meaning they're two, like, so this whole thing right here it's called a phosphodiester bond.
Wait, but is this the same ester as before?
Not really, no. Really, I mean, technically they're like, there are O's here. There's an O like this, and then there's like an O. There's like an O here, like that.
That's fun.
But anyway, those are called phosphodiester bonds, right?
What's a dicleotide?
A dinucleotide means just like 2 nucleotides together, just like a disaccharide, right? Okay, so this is pretty easy. And I would keep, if I have a polymer, I just keep adding to the 3 prime end, right? Oh.
That in turn can be called an oligonucleotide.
Yes. Yes. Very good. And if I have more than 10, I could call it polynucleotide. All right. Yeah. Or then it becomes, nucleic acid, right? Okay. So do you guys notice? I want you to not look at the bases for a second. I want you to look at this part. This is what? Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, right? Those form what they call the backbone of the nucleotide, of DNA, right? So that's the backbone. And this part kind of goes, these go in the center, right? So those are the bases that are extending toward the center, right? Do you guys know what the name of the documentary thing that yesterday was called? It was called it was called the double helix, right? How many polymers, how many strands do I have here?
Uno.
I have just one, right? But in order to make it a double helix, what do I need? I need another one, right? So here, I think this is gonna work. Let's see if this animation works. Okay. What's that again? Nucleotide. Where do I add it? Where am I going to add my next one? 3 prime, right? 3 prime water each time, right? All right. So there, I have my little my back. Here's my backbone. The base is not part of the backbone, right? Sugar phosphate backbone. backbone, and then there's like all those phosphodiester bonds. But if I want to connect this with another strand to make the double helix, I have to, the other strand is oriented it's upside down from this one, right? So, what's that? Do you guys see how it's a nucleotide, but it's like totally upside down, right? So there's a nucleotide, and where am I going to add on my second nucleotide to this one? Onto the 3 carbon, right, which is where that hydroxyl group is. So I'm going to add it here. And what comes out each time? A drip of water each time, right? Yes.
So out of this, like, what's the TA and what's the CG?
You'll see. We'll get there. All right. So Mike, tell me where on this strand over here, which is the 5' end and which is the 3' end?
5' end is with all the red dots and the. Oh wait, no, wait.
There's only one 5' end.
At the top.
At the top, right? And what's good? And what's the thing that is at the 5? The very 5' most thing is a phosphate group, and the very 3' most thing is going to be a hydroxyl group on the deoxyribose molecule, right? Lidu, what about this molecule right here? Which is the 5'?
The 5' would be the phosphate.
Okay, so where?
So that would be at the bottom.
At the bottom, right? So this is the 5' end of this one, and then we're building up toward the 3' end, right? So this arrangement, is it like, are they parallel? No, it's actually described as it's described as, okay, you're right, but not quite yet. They're described as anti-parallel because they're running in opposite ends. Are they, like, so this one ends in a OH? Are these going to be like mirror reflections of each other? No, they're not going to be. So this is going to be the same as that one, and that one's going to be the same as that, right? So they're kind of like, this one's going to have a phosphate, that one's going to have a phosphate, that one's going to have with OH, that one's going to have with OH. Right? Okay, so this is called anti-parallel, right? And then once we have this sort of ladder going on here, then that's when the helix forms. It starts to twist around itself like that, beyond that. That's where the helix thing comes from, okay? All right, so the question is, how do we hold these 2 strands together to each other? Bonds, what kind of bonds? They're hydrogen bonds, right? Remember in the beginning when we started talking about hydrogen bonds? And I said hydrogen bonds, what did I compare them to?
Oh, you said it was
the toddlers, right? So relatively weak, but again, they're very, very important, and they hold together important molecules like cellulose and DNA. Okay, so those are hydrogen bonds. All right, so we have the backbone down. You guys are you have the framework. Now we're going to get into the ATCG stuff that Mike was asking about, right? All right, so basically we have all these things, the bases. We've been ignoring them right now, but let's get into it, okay? So there are 2 main classifications of bases, right? One's called the purines, right, which have 9 members in their rings, right? And 9 members. How many rings are there here?
2.
There are 2 actual rings, right? So here's one ring, and here's the other ring. Where do I get the 9 members from?
Carbons.
How many members are
Number
there?
of non-adjacent sides or
Yeah.
carbons or whatever.
So the number of, they're not all carbons, some of them are nitrogen, but the number of points here, right? So there are 9 atoms that are all kind of contributing to making that 2-ring structure, right? Okay, so those are the purines, and then there are ones called pyrimidines, which are only consisting of how many rings here?
6.
Not 6 rings,
One.
1 ring, but 6 participants in that ring, right? So this is a double 9-membered ring, right? And this is a single 6-membered ring, right? Do you guys know what the 2 purines are? And the way that I remember it, I think of 9, right? 9 members. So the 2 ones that are purines are going to end in 9. Exactly, good. Adenine, I think it's pronounced adenine, but if 9 helps you remember it, adenine, and then guanine.
Reminds me of my friend Yenny.
What?
Reminds me of my friend Yenny. Her name is Yenny.
Yanin, like instead of like Janine, but it's with like a.
Yeah, it's well like instead of Y, it's Y.
Oh, that's what the AC.
Oh, Yanin, like YNIN.
See, like my 7th grade science teacher.
Was what?
Never mind.
You have to say it.
My 7th grade science teacher was so bad. She never explained this to us. She just said, "Oh yeah, it's AC."
And then our teacher that literally tortured us.
Well, I mean, that's just an easy way to remember it, but there's not like a rule for it. Like, there's no actual. That's just like the way I remember it.
No, our teacher tortured us with it.
All right. And then the pyrimidines are the other two. And why did I underline the Y, I think? Because the other two have Y. The other two have Y's in them. So thymine and cytosine, right?
No.
Right?
Science.
It's just a way to remember. There's actually no science involved in those mnemonic devices at all. Okay. So if I ask you what the purines are, you say adenine and guanine. If I ask you what the pyrimidines are, you say thymine, cytosine. All right? Okay. But there are rules for which ones can pair with each other, right? And that's called complementary base pairing, right? Complementary. Do you guys know what complementary means? Yeah. So they they add up together, or they make like a complete sort of thing together, right? So they actually mentioned this guy yesterday. His name was Erwin Chargaff, right? He's like this French guy who is measuring like all these samples of animal stuff, and he found out, and he didn't actually put it together, but he found out like, wow, in all these different animals and all these different organisms, this like they all have the same amount of A and T, right? And they all have the same amount of G and C, right? So again, Watson and Crick took that information, and what did they do?
Stole it.
They stole it. I don't know who was like so, bad in this case, but yeah. So because Chargaff didn't know, he didn't realize. But anyway, so then they said, okay, A pairs with T and G pairs with C. What do you guys notice in terms of like the types of rings that A is and T is?
Complementary.
Like a 9-membered ring.
This is a 9-membered ring or a double ring, and this is a single ring, right? And so this is a purine, and this is a pyrimidine, right? And this one is a purine, and this is a pyrimidine, right? And why does that sort of make sense that they have to pair with each other?
Because then they'll be like same length.
So then, like, the whole molecule itself will be the same, not length, but width. It'll be the same width, right? Because if I try, suppose I have, like, so here's my sugar phosphate, like, backbone, let's say, right? This is, like, such a bad drawing, right? And then suppose I have a base here that's 2 rings, right? And then here's my other sugar phosphate backbone that's going, again, so this one's going this way and this one's going anti-parallel, right? Suppose I have another one that I'm trying to fit two rings in here, right? And then this one only has 1 and 1. Is that going to be the same like width consistently? No, right? So I need to, whether, it doesn't matter which side the 2-ring one is on, right? So I can have the 2-ring one over on this side, right? But as long as I have the other one, so as long as like there's like a total of 3, like the width of 3 for each base pair, right? George?
My teacher did teach me a way to remember like which one goes with what. The apple goes in the tree, and then the card goes in the garage.
Okay.
And then car goes in the
Americans
garage.
are trash, Germans are cool.
The garage
Let's
has
be honest, it's just that.
let's see with G.
Let's be honest, it's just that. I don't know, it's just the Austrians are trash.
All right. So this is a really good way. And, that's a really good way to remember it too, because it does help.
March Madness sells basketball.
Just order matter. Like,
I just remember as
Austrians are trash, Germans are cool.
I just remember as TACG.
Whatever way helps you. That's fine. We do.
just order matter.
What do you mean?
Like if I have A pairs with T and then
Yeah, that's fine. It doesn't matter. Order doesn't matter. All right, so that's a good way to remember it. But you need to know that there's always going to be a purine with a pyrimidine, right? And the reason is because you want the width of the DNA molecule to be consistent throughout. Okay? So I might give you a problem like this on a quiz. Alright, so this is based on Chargaff's rule. You have to know what Chargaff did, right? He measured all these samples, right? And so in a given nucleotide sample, the percentage of guanine is found to be 20%. What are the percentages of all the other bases? Think about it before you do it. Just write it down, right? And I want everyone to have a chance to like really think about how they might solve that.
Wait, that's not bad.
Huh?
If I'm right, then that's not bad. But if I'm wrong, then that's really funny.
It's pretty. I mean, it takes a little bit of reasoning here, but it's not like impossible. It's like kind of common sense. You can arrive at it based on what I've given you already. So you should be able to figure it out. All right, I'm going to call on someone who doesn't usually volunteer. I think maybe we'll go with Anthony.
I think I got it.
So what do you think? And tell us your reasoning behind it.
There's 20% in the cytosine.
Oh, okay, there's 20% in cytosine. How do we know that?
Because they're the same amount.
Why are they the same amount? How do we know that? Because what are they doing with each other in the DNA molecule?
Pairing.
They're pairing together, right? So we know they're going to be 20 here. So how much will be in the other two then?
80%.
Huh?
80.
80? 30 in each? Or 30 in which one?
Both.
30 in both, right? How do we do that? Because you know, it all has to add up to what? 100, right? So if you have 20 and 20, right, that takes out how much from 100?
I didn't know how.
Right? That means that so this is 40 total, right? So there's 60% left, right? But we know that adenine and thymine have to be equal, and because they're pairing with each other. So then what do we do? So this is going to be 30, and this is going to be 32. Right? Really easy? That was on the PSAT?
No,
Oh.
but that was like the easiest thing ever. I was just like sitting there like.
This is the easiest thing ever?
No,
the
the
PSAT.
PSAT.
Oh.
The first math section was about like 2 minutes long.
You guys are just too smart then.
It was 8 to 9, but it's like bottom level of 8th grade. So most of us took it in like 6th grade.
You guys are amazing. All right. Yes. All right, hon. I have a question. Arsh, Hannah has the floor. All right.
So, like A was like paired with like C or G. Would that cause like a mutation?
Yes, and that does happen sometimes. So like by accident, sometimes like our and we'll talk about this when we talk about replication. And but basically DNA is really good at copying itself, right? So but sometimes by accident it will put the wrong base in there, and then that causes problems later down the line. Yeah.
Like red hair.
Or it causes things like the ability to digest lactose for your whole life, which is a good mutation because otherwise you guys wouldn't have like ice cream or like milk or.
Wait, not being lactose intolerant is a genetic mutation?
Yes, originally most people like you're not supposed to be able to digest lactose as an adult. Like children, obviously they're drinking milk, so they babies. But then after that, you're not supposed to be able to drink milk. But we mutated, and now we can digest milk, and so it's great. Yeah, but some people don't, and I think most of the world is lactose intolerant still.
Wait, could one child be lactose intolerant if the other not?
Yes.
Yeah. It's a gene.
Like, I want the
So.
exact same as.
All right. Shh. Okay. So, question: nucleotides, adjacent nucleotides, so nucleotide monomers, are joined together through what kind of bonds? Phosphodiester bonds. Okay, I mean adjacent monomers, right? So they're not attached by hydrogen bonds. You guys know that, right?
I just read hydrogen.
Okay. Okay.
Let's talk about the bonds.
Okay, phosphodiester, and then base pairs are bonded to each other. There we go. Okay. All right. Don't scare me again. Okay.
Wait, no.
April Fools.
What?
Should I finish it?
Yeah. Go ahead.
Yo, Omar.
All right.
Omar came up with a brilliant idea to contain the virus by sneezing into your own shirt.
If you don't have a tissue, it's better than sneezing on your arm. Because
Yeah, you're keeping it inside
not
your.
many people come into contact with the inside of your shirt more than that.
It's what it is.
No, not wrong. You're right. You should like patent that idea. Other people steal it because
It's like a reusable tissue thing to use like.
Well, for you, I think it's what.
it could go viral and people could.
Oh, get it viral? I like that.
Viral. All right. All right. So I want you guys to take a look at this little depiction here. All right. So first of all, what is this thing that's selected right here? What's that thing that's highlighted right there?
It's the nucleotide.
That's a nucleotide, right?
It's in the basis.
Because it has a.
Oh,
I'm sure
so
you're
that's
right.
that's the nitrogen.
A phosphorus and a base.
Yes, the nitrogenous base is one of those four bases, right? So in the beginning, just for simplicity's sake, I was just putting base. But now you know that it could be any one of those four. It could be one of these, which these two are what? Purines. And these two are the pyrimidines, right? So this is a nucleotide, right? What is this bond here called? So that group itself is called the phosphate group, and this is the phosphate group. Right? But the bond, so this bond, including this bond over here and this bond over here, that's a phosphodiester bond, and that's connecting these two, this nucleotide to that nucleotide, right? So things that connect nucleotides to each other into a polymer are phosphodiester bonds, whereas these bonds right here, those are the hydrogen bonds, right?
Yeah.
I want you guys to notice the number of hydrogen bonds between each of the different like base pairs. Between A and T, how many hydrogen bonds are there?
AT2 GCP.
So you guys have to know that, right? Do you see how they're? All right. So between A and T there are 2, and then between G and C there are 3 hydrogen bonds, right? And
G86 cars.
these refer to the hydrogen bonds, right?
T2, G82.
Two apples in the tree.
And 3 cars in the garage.
3-car garage.
Genius.
All right.
Two apples.
Alright.
3-car garage seems excessive, don't you think?
Alright, 2 off these are trash, and 2 of those are cool.
I as medical examiner, I want to have 3 cars.
You wanna have 3 cars?
I want to have a Porsche Cayenne. And I want to have a
The
Mercedes
G-Bag.
Benz.
Oh my god,
And
you guys are the ones the
Wow, very specific. You've thought a lot about this already. That's good.
I have a really involved one.
And so you'll have a 3-car garage. Okay, that's fine.
I kind of like the C-Class sedans. With the hatchback.
All right. Anyway, all right. So we're good with this. We're good with this. Good. That's great.
2 off these are trash.
All right. All right. We're almost done. We're really close. All right. So Rosalind Franklin, right? You guys, her contribution to this whole thing was. So she knew that it was a helix because she used what technique?
X-ray diffraction.
It was called, so it's called X-ray diffraction, or they call it X-ray crystallography, right? And it makes this like pattern. So basically, like you're shining this light at this thing, and it makes like an opposite pattern from what like the actual way it's oriented. You don't need to know the specifics about that, but basically there is this photo 51, right? And that showed this like perfect, like the measurements of that. Crick was able to, remember he was like sketching it ahead of time and he was kind of like predicting what it would be. And based on her picture, he was able to conclude that it was indeed a double helix, where the bases were oriented in toward, remember there was a while where they were kind of like playing around with the models where the bases were kind of flying out and it was like growing and growing. Yeah, but so actually the bases are facing inside. And so there are these twisted strands and the bases are inside. So it looks kind of like this if we were to show it, right? See how it's like sugar, phosphate, sugar, phosphate, sugar, phosphate. And then again, the base is not coming off the phosphate, it's coming off of the sugar, right? What carbon of the sugar is the base coming off of again?
What carbon of the sugar?
Which carbon of the
3 prime.
the sugar, I mean the base is coming off of the 1 prime, right? Whereas the hydroxyl group is off of the 3, and then the phosphate is coming off of the 5, right? Okay. So yeah, do you notice again, here it is, so here's AT, there are 2 bonds, 2 hydrogen bonds, and then GC has 3 hydrogen bonds between them, right? Who knew like all this stuff before? Are you guys like, alright.
I didn't know about the hydrogen.
Okay.
Wait, let me go back. I knew it did.
Which one?
Except ATs.
Can you please
Okay.
I'll post this I'll post it like right now. If you guys don't distract me, I'll put it like on Classroom right now.
Okay, you can go with steal kids' fidget spinners and hang them off the ceiling.
Ceiling fan time.
She was sucked on it, bro. Her name was Mrs. I'm not gonna name drop.
Alright, I have a question here. So, those are phosphate groups here. I want you guys to just kind of take a look at this. Shows a little bit about how the actual dehydrogenation synthesis is happening. You guys don't have to know this. You just have to know that there's a phosphate group that makes this phosphodiester bond. But I want you to look at the phosphate group itself. What does it have a lot of? Oxygen. It has a lot of oxygen, and it's actually negatively charged, right? So overall, DNA is negatively charged, right? And then if you were to put DNA in an electrophoresis apparatus, right? Where should we put the wells? Do you think we should put the wells right in the center?
Positive.
So we should probably put the wells closer to the...
No, negative side, no.
So instead I would put the wells closer to the negative side so that the DNA, I would put them in the wells, and then the DNA would travel toward the positive electrode because it is negatively charged itself, right? Yes.
Is the DNA
Just trash.
full negative charge or is it partially negative?
It's an actual negative charge because this oxygen has an extra electron. So it's actually an ion. So yeah. So it's actually negatively charged. Yeah.
When DNA is replicating itself, doesn't it unzip and then attach to it?
Yes, we'll get into that all next week. Yeah. This week, yeah. But you wrote something about helicase and that all involved with RNA.
Transcription.
But helicase is part of DNA replication.
Oh.
RNA transcription is something totally different.
I know, so
But it was good. You guys were good. All right, I'm gonna pass back your quizzes real quick. Do you think we have enough time to look at them?
I have the best way to remember it. All right, so you have 2.
All right, listen. Tomorrow I'll show you guys the quizzes, and I want you in the first few minutes of what? What? You want to look at it now?
Yeah.
I think that there are some people who have more questions wrong, those who might need more time to look. So tomorrow we're gonna do that first, right? And then you guys are gonna do your models. You're gonna make models. I'm just gonna pick groups out of a hat. And then I'm gonna give you all these supplies, and you just make your own models, and I'll give you instructions. But it's free, anything goes.
I like making models.
They're not with these kits though. They're with random little beads that I stole from my daughters. So.
Well, it's not because she was killing rats.
Alright guys, I made an analogy, you can easily remember. 2 Austrians are trash, 3 Germans are fools. You have 2 AC and 3 GC. 2 AT. And the 2 Austrians are Addie and George.
I passed down my ability to kill rats to my daughter. Okay.
So now you have the 2 AT Austrians are Addie and George. Addie and George, AG, adenine, guanine. Those are the 2 purines. Done.
I gave up an 8 on
I know.